Hi everyone, Jason here at the Whittier Public Library with another STEAM-powered science take-home science kit. Since March is Women's History Month, today we are going to build a model of DNA, which is probably the most important molecule inside your body. And we're going to learn a little bit about Rosalind Franklin, who was an English scientist who made some discoveries that were very important to discovering DNA, its structure, what it's made of, and what its shape is. Okay, let's go ahead and get started. So in your kit, you should have a couple of red vines. We have extra long ones here, and we've given you a couple extras in case they break. You should have some of these little mini craft dowels, which are sort of like toothpicks, but without points on the ends. We tried using toothpicks, but they were really sharp, and we kept poking our fingers, so we're using these instead. You should also have about 20 gummy bears. You may not use them all, but they're there in case you need them. And what you're going to want to do before you start is you're going to want to look at your 4 colors of gummy bears, and you're going to want to assign them letters. The letters are going to be A, T, C, and G. Make sure that there's one letter for each of those colors, or one color for each of those letters, rather. And what you're going to be doing when you're building your DNA model is you're going to make sure to always pair A with T and always pair C with G, because that's the way it is in real DNA. Okay, so let's go ahead and build our model. So remember, we're going to have 4 colors of gummy bears, and we're going to designate one of them as A, one as T, one as C, and one as G. Now we're always going to pair up A with T and C with G. So what we're going to do is we're going to take one of our little dowels. These are sort of like toothpicks with no points on the ends. And we're going to take one of our gummy bears. Let's take an A. And we're going to push it through. You might have to spin it and twist it a little bit to get it through there, but you should be able to get it through just fine. And then we're going to take a T, and we're going to put it on the other end of that. Twist, twist, twist. You can kind of hold it and push it down a little bit. Just be careful with these so you don't poke your fingers. That's why we didn't give you toothpicks, because they're kind of sharp on the ends. Okay, so we're going to make another skewer. We're going to do a C and a G. So green will be our C. Remember, just twist and spin this to get it through. Push down on it a little bit, maybe. There's a C. Stretch it out so it covers more. And then a G. And push that through. We're going to push down a little to get it through. Okay. Now we're going to keep doing until we've done all our skewers, but I'll show you that in a minute. Okay, now we're going to take one of these. We're going to get our 2 strands of our red licorice, and we're going to push it through maybe just like a 1/2 inch or so from the end, about there maybe. So we're going to push that through there. It's a little hard to get through, so what you might want to do is do this. Or another way you can do it is like this. You can push it down and push it down through there, like that. Flip it over. Push, push, push to pop it through. It's a little hard, but you'll get it. Okay, so that's going to be our first one. These are called base pairs. That's our A and T. We have our C and G right here. So we're going to space these maybe a little bit more than a gummy bear length apart, like right about, let's say right about there. And you can use that gummy bear to measure and get it in about the right place. It doesn't matter if they're exactly spaced, but try to space them equally apart to have a more realistic DNA model. So I'm pushing down. I'm going to push that through. Do this one at the top. I'm going to push that through. Try and keep them as straight as you can. It's not going to be perfect, but as close as you can. Okay, now I've got my second base pair. Now I'm going to continue, and I'm going to go to quicker motion so that you can see this get done completely. Okay, so our model is now built. Next thing we're going to do is we're going to turn it into the shape or structure of DNA, which is called a double helix. And a helix is basically a spiral. So what we're going to do is we're going to take both ends and grab it with our hands like that. Twist your hands up a little bit so you can do this. And we're going to give it a good couple twists. And now we have something that looks like the shape and structure of DNA. Okay, so let's talk a little bit about what we made here today. So this is a model of DNA, which is short for deoxyribonucleic acid. And this is probably the most important chemical or molecule in your body. What this is basically the instructions to build you. So for example, if you ever bought a toy or a piece of furniture your family bought and there's a little sheet of instructions in there, you have to put it together yourself. That's what DNA is for you. It's that sheet of instructions that tells you how to assemble that. It tells you how to assemble you. Now, we have about 30 to 40 trillion cells in our body, and every single one of them has a copy of those instructions. So there are lots of copies of DNA in your body. Now, this, and it is made up of these things called base pairs. And what these are basically the code, the order that these are in, or the code that's specific to you. It's called your genome, and it tells how to make you and only you. Now, one other thing, this model we made is of course much bigger than real DNA. It's in fact, it's probably more than 50 million times the size of real DNA. But it's also a lot smaller. In this particular model I made, we have 9 base pairs. You might have 8 or 10, but our real DNA has about 3.2 billion base pairs. So this is a very, very small section of our DNA. Okay, so let's talk a little bit about our scientist, Rosalind Franklin. About 70 years ago, there were lots of scientists studying DNA. We didn't really know exactly what it was, what its shape was, what it was made out of, what it looked like. And the problem was it's so tiny, you can't really look at it. Even with a microscope, you can't see it. So we needed a new way to find out what it looked like. Rosalind Franklin was an expert in a technique called X-ray crystallography. And X-rays are what doctors and dentists use to look at your bones and teeth. But they can also be used to take pictures of things that are extremely tiny. So they were perfect for taking pictures of DNA. You remember, this is about 50 million times the size of DNA, so DNA is very, very small. But Rosalind Franklin, in 1952, was able to take a photograph. It was known as photograph 51, that gave a pretty good idea of what the shape of DNA was. This photograph was shared with 2 other scientists named James Watson and Francis Crick. They eventually used that photograph and a lot of other clues. It was a key clue to figure out the shape and structure of DNA, what DNA was made out of, what it looked like. At the time, Rosalind Franklin unfortunately didn't get enough credit for being part of this discovery. But later on, over the years, she's become very famous and come to be recognized as one of the important scientists in the discovery of the structure of DNA. And if you want to learn more about her, we do have some books at the library now that you can read and find out a little bit more about her. So that's about it for today. Thanks for joining us to build a model of DNA. If you'd like to learn more about any of the topics we talked about today, we have a lot of books at the library that you can come by and check out. Here's some examples over here. Come see us soon at the library, and thanks for joining us. Goodbye.